in the Southeast Asian countries. So when we talk about their applications, so the polyphenols are the excellent antioxidant agents. So this is the like their exponential property, that is the antioxidant. They can prevent or they can inhibit the generation of free radical into the any system. Either it can be in the human body system or in the food system. So antioxidants are the substance that can prevent or slow down the damage to the cell caused by the free radical. What happened? Why free radicals are generated? So in the human bodies, the free radicals are mainly generated due to the stress that can be due to the exposure, too much exposure to the sun. So these are the conditions that can generate the free radical. So what these free radical they do, they will take electron or the atom, a hydrogen atom from the other healthy atom, and they will make it as a free radical. So they continuously they will produce a free radical. This is their behavior. So what happened when we add any antioxidant? So antioxidant have ability to donate electron or a hydrogen atom to the free radical, so they can make it stable. Yeah, like in this figure, you can see that they have that the antioxidant is giving electron to the free radical, so that then it will make it stable. So it will be not able to uh, execute an electron from another healthy atom. So why these polyphenols have a high antioxidant? As you can see that this is a like example of kind of hydroxyl or hydrogen atom. So they have the ability to donate their hydrogen atom or the electrons to the free radicals, so they can make them stable, a healthy atom. Okay, so this is what I was talking about, the stress, oxidative stress. These are, they will lead to the produce of the radicals that can be reactive oxygen species or active nitrogen species. So those, they can bind to the proteins of the our cellular system or the lipids or DNA. So once they bind with the any protein or the DNA of the cellular system, so they can extract hydrogen or an atom from electron from those proteins or the lipids, then they can make them a macro radical. So this is how they can change the structure of a protein or the lipid. So once you know that when we have a change in the structure of a proteins, its function will be changed. For example, if some protein are known to produce the cell, a new cell. So once those proteins are degraded, they cannot make a new cell. So that can lead to the aging. There will be a no formation of the new cells that can cause a old cell formation, and there will be no formation of the new cell. So that will lead to the aging. Other than this, there are other functional, I already talked about the like hypertension and obese. Those things can be happen due to the impaired system, like damage of the DNA or the oxidation of the protein. So when we talk about the application of polyphenols in the foods, so foods are, we know that they're mainly rich in the proteins, as well as some like fatty fishes, they are rich in the oil. So those radicals or those oxidation of these compound like lipids or the protein, they can change the functional property of a food, or they can change the appearance of the food, particular food, as well as odor. They can produce the odor into the food, so that will lead to their reduction. So to inhibit the oxidation of lipid, we need a polyphenol or an antioxidant that can inhibit the oxidation of these protein. Other than the antioxidant activity, the these polyphenols are known to possess antimicrobial agent, like they can inhibit the growth of the various bacteria. So in overall, I can say that when we want to preserve a food for the longer term, so like you can know that now the life speed is very fast, so everyone need ready to eat. So nowadays we are focusing more toward the ready to eat foods. So that therefore we need some food that can at least keep for 1 week or maybe 2 weeks. So to preserve any food that is rich in the protein or the lipid, we need the application of the antioxidant. So that how we can inhibit the oxidation of proteins as well as the lipid. Moreover, if oxidation of lipids or protein occur, so they can lead to the production of bacteria like microbial spoilage, because these oxidized or hydrolyzed lipid or proteins will act as a substrate or food for the bacteria to grow. So we need some additives that can inhibit microbial spoilage also. So considering the polyphenols having both applications, so we can add into these polyphenols into the food, and we can enhance their shelf life. Shelf life means how long we can keep that food without any spoilage. So polyphenol can increase the shelf life of a food. Now another application of polyphenols, like because of their special structure, you can see that these are the because of the hydroxyl presence of hydroxyl group and the benzene, they can act as a conjugate between the different biomolecule. For example, this is a one protein and this one is another protein. So they can conjugate between, they can bind with these proteins. They can act as a bridge. As you can see that this is a polyphenols, and it can act as a bridge between the two uh, two proteins. So this way they can enhance the application of the protein. For example, if you want to add any protein additive into a food, but that protein has low antimicrobial activity as well as the low antioxidant activity. So we can conjugate these polyphenols with the proteins, then it can increase their activity synergetically, because polyphenol will also provide its antioxidant or antimicrobial activity, as well as protein also have some kind of antioxidant or antimicrobial activity. So in this way, we can increase the activity of proteins. Similarly, other than the protein, we can produce chitosan or conjugate. These are the chitosan is a polysaccharides we usually obtain from the shell of the crustaceans. For example, shrimp shell. So these are a kind of polysaccharide, but they are not soluble in the water. So when we combine with the polyphenols, they become a water soluble. So you can see that it can increase its functional activity also. So once any compound is soluble, it can act as its bioactivity will be increased. So this is the another application of conjugate that we in our lab we do to increase the antioxidant of protein or the polysaccharide. Why? Because the polyphenols already have large number of hydroxyl groups, so they can add more hydroxyl group into the our biomolecule, such as protein or polysaccharides. So I will be not uh, discussing about these mechanisms, so we don't need to focus on this one, but this is the general, like how we produce chitosan conjugate with the phenolic acids. So here you can see that. So when we combine, these are the different polyphenols or the phenolic acid that we have already combined with the protein and the chitosan. So we found that their antimicrobial activities increase, carcinogenic activity also increase, as well as the allergic or anti-aging activity also increase. But we have found profound increase in the antioxidant activity. So this is our main motive to conjugate the proteins with the polyphenols, because we want compound that have high antioxidant and antimicrobial activity. So why we need preservation of the seafood? So like we are already discussed about the application of polyphenol that can be used into the preservation of food, especially seafood. So we know that the fish and other proteins like fish and this is the shrimp, these all are spoiled product. So you can see that once they are captured from the sea or from the rivers, their preservation we need to done their preservation. If they are not stored properly, they are easy to degraded or spoiled within a 1 day. Even you see the next day, if you haven't keep these samples in ice or refrigerator, they will spoil very next day, because they are rich in the vitamins, proteins, and minerals, as well as the PUFA. This is the polyunsaturated fatty acids. So these are the compound that can cause their spoilage very fast. Once what happened? So like in their digestive system, usually what happened when you see fillet like this is a fish fillet, like a fish meat. These are the sterile, means they don't have any microbial activity or other things. But when you keep the whole fish for a longer time, the enzymes or the bacteria from their digestive tract will come to these muscle. So that's how they can cause their spoilage. So usually what we do, so once we catch the fish, we remove their digestive tract to inhibit the enzymatic activity and the filtration. Once we have our food like a fish fillet, the fresh one, we want it to keep for a longer time. So we need to preserve using different kind of technologies, as well as the use of different kind of additives. So to solve the problem of the spoilage, there has been a wide number of chemical synthetic preservative like BHA and BHT has been used, but they have serious health human, like they cause the serious problems in the human, such as they can cause cancer. There are some reports has been reported when you use these synthetic preservative at a little bit higher amount, they can cause cancer to the human. So that's why and preservative that is from the nature. So plant extract any bark or fruits, like there are some fruits that has been not used for the human consumption. So those are rich in the polyphenols. We can use those fruits for the extraction of the polyphenols. Moreover, they are rich in the essential oils. So essential oils has been also used for the preservation of food. So these are the some plants that we have been using in our lab, like guava leaf, jammuang leaf. This is a like typical Thai native plant, and the betel leaves, as well as the green tea. So these are the plants that we have been used. But the problem with them is their green color. Whenever we extract polyphenols from these plants, they have a dark green color. So once you know that there is a fish and you added your polyphenol extract and it look like a green, so no one will buy that one. So we need to dechlorophyllize that extract. So that to remove the the chlorophyll using a green process. Green process means I will discuss later about this one, sedimentation process. So other than those leaves and these, we have been using like agriculture waste. Like in the Thailand, the coconut is widely consumed and is you can see its consumption in the Thailand is too much, even in the Thailand, Malaysia, and neighboring country. So these coconut husks, we extract polyphenols from these waste using ethanol, like 60% of ethanol that has a high antioxidant and antimicrobial activities. So here we come to the separation of the chlorophyll from the our plant extracts. So what happened here? One, I told you that it can change the color of any food whenever you add this one. And another thing is that they can act as a oxidizing agent, means they can enhance the oxidation of the food. So therefore, we need to remove chlorophyll from our plant extracts. So there are some traditional method in which we use a solvent, for example, hexane, chloroform. These are the solvent we have been using, but because of the FDA regulation, we cannot use because they are toxic. So there are other technologies that we can use for the extraction of the chlorophyll from the plant extract, but those are the wild like expensive and it's not feasible to use. Another, the that we have a developed method, this is already developed but we are like modify this method. Just use of the water, we can separate our extract from the chlorophyll. So what we do first, we just dissolve our extract with the ethanol, and then we freeze dry that part. We make a powder of that ethanol extract, and then that powder is dissolved into the water at a different ratios. So you can see, and then you can keep it for like 24 hour at 4 degrees centigrade, and you will get the chlorophyll will be separated at the base, and the upper will be the clear extract without the chlorophyll. You can see that this is our beetle extracts. So you can see how much is dark green in color, and when we use those different methods, so you can see that the green color is almost gone. Actually, we cannot remove exactly all the chlorophyll, but most of around 80 to 90% has been removed using this sedimentation process. The this one I would like to discuss about the it is a kind of nanotechnology uh, for the for enhance the activity of our polyphenol extract. So once we get a powder a longer time, and even you keep in the dark in the light, it will oxidize. This compound will denature, or not denature, the activity of this compound will be lowered if you keep as such. So to preserve its activity, what we do, we have done encapsulation. So what we do, we uh, you can see that these are the two layers of compound, and we store our sample here, this one. So but what will happen, your sample will be here and it will be protected by the two layers. That is called encapsulation. So liposomal encapsulation. In this, what we use a phosphate, uh, we use soya phosphatidylcholine in this one. So what it will do, it will encapsulate your compound inside, and it can protect from the outer environment. So one another thing, and it will increase its releasing efficiency also. So for example, you can add directly to the substrate. So once these liposomal will bind here, so it will be easy for these compound to release out into the cellular system of any food. They can also lower the green color of our the green extract, the polyphenol extract, as well as they increase their activity also and their activity. Moreover, it can like because polyphenols have very strong color order, so they can also mask their order. So once it's here, but when it go into the system, so these walls will be hydrolyzed and this compound will come out, so that can it can show its activity. Here you can see that. So what we did, we use ethanolic coconut husk extract that I show you last slides. So these are the compounds. These are the nano liposomes. So the our ethanolic extracts is inside them. So this is how we increase their stability as well as their activity. And this is like a micro SEM structure is almost around 10,000x. This one, yeah, I think almost 10,000x. Now, we coming to this application part. So, the shrimp become dark because of the polyphenol oxidase activities. This is a, like a melanin kind of product. So this will cause a blackening of the shrimp. So, but when we use catechin and EGCG derivatives and treated these shrimp with these compounds, so they can inhibit their formation of dark color, as you can see that, because of their antioxidant activities. And moreover, what when we use ultrasound is like it will produce a wave like this, so that can produce a small bubble inside a cell. So once the wave is going increasing and increasing, those bubble will burst out. They will be broken into the pieces, and then they can cause a pore inside the cell. So you can see that these are the shell. They are very hard. So it is very difficult for catechin or other polyphenols to enter into the cephalothorax. This is actually head we call cephalothorax. So it's difficult for those extract to penetrate into their system. But when we make a pore using ultrasound, so they can easily penetrate deep into the cephalothorax or the head of the shrimp. So then they can show a more activity. So these are the examples of combination of new novel technology with the our extracts. This one is a fish ball. It's a like a jelly product widely made up of fish means. These are the so they are gel like structure. So what happened? We need the elastic, the rubbery structure of these walls. But what happened when you use only fish means? When you use only fish means, so they have okay what, and that will be like this one. So in these pockets you will get the water. So what happened when these my proteins have low gel forming ability, so they make a gel like this one, not regular. So you can see that these are the large holes, somewhere very large hole, for example this one and this one. But in this, the order structure, you can see that somewhat like this one, small hole, but order structure in this case. So polyphenols, as I show you in the last slide, here polyphenol, it will protein that polyphenol, it will bind to this protein and this protein, and this is how it can decrease this whole to this one. You can see that these are the formation of bonding between the proteins. So these all are because we add tannic acid here. So once we add tannic acid with increasing concentration, so you can see that these holes are decreasing. See, and this one. So once these holes are increasing, so they can hold a more water inside the gel. So it will make a juicy and elastic structure. So that's how the another application of polyphenols we can use to improve the functional property of food. Like it will look also good and it will be darker, lighter in the color, and texture will be very good. As I was talking about the antimicrobial kind of proof, what happened? So our polyphenol, they can cause a leak hole, so it can lead to the cell death. This is without the, sorry, uh, the bacterial cell without treatment of polyphenol. But when we treat with the polyphenols, so the cell dead. This is another cell, Listeria monocytatis. It is also one of the bacteria that can cause the spoilage and as well as the human health problems. So you can see that they all are dead. And the shape of the cell also was not proper. This is the normal cell, it's healthy cell. But when we treat with the polyphenols, so that they become flaccid. Here is another example of E. coli and Pseudomonas aeruginosa. You can see the cell death. So this is how the activity of antimicrobial. What they do, they will bind to the bacterial surface and then they will interact with the peptidoglycan or layer of the protein or the polysaccharide lipid walls at the bacterial cell. So they interact and then they cause the leakage of the cell. Or so once it leaks, so other polyphenols will penetrate inside the cell. They will go inside the cell and they will uh, inhibit the DNA translation or the transcription, those protein synthesis. So that's how they can inhibit like cellular metabolism of the cell, bacterial cell. So they that can cause their death. So this is another example. Maybe all of you know about the sushi and the sashimi. So this is the tuna fish and this is a salmon, orange color. But when we keep at a fridge, even you keep in the fridge, they become turned to the dark color. So because of the oxidation of the myoglobin. Myoglobin is responsible for the red color of an orange. Orange color is because of the astaxanthin also, but this red color is because of the myoglobin red pigment. So you can see that this is a controlled tuna sample. So you keep it for a long time, it will be like dark like this one. So, but when we make a film, like make a pouch, or in which we have cradone extract. This is also a local plant added with the catechin. It is also polyphenols. So when we add into the film, this pouch is like made up of the protein. So that pouch was protein was added with this extract. So when we wrap these fish pieces here inside this one like this, and this one is a control film, like without catechin and those polyphenols. So I will say polyphenol. This is without polyphenol film wrap, but without polyphenol. This is the fish myofibril protein. Only fish myofibril protein incorporated with the catechin and the cradle. So in this, they add this polyphenol extract, and this is a normal film that has been used for the packaging of food. So you can see that they can at least keep until the 6th day. The red color is still here because of the presence of the polyphenol. So polyphenol will inhibit the auto oxidation of the myoglobin. So that's why it can keep red color for the longer period than other samples. So this is another example of having polyphenol with antioxidant activity. So when we come to the human health improvement, so we have done in vitro antioxidant studies, anti-inflammatory activity, and anti-obesity activity, as well as the wound healing activities. So what we did, we have our phyto-oligosaccharide. This is also kind of polysaccharide and hydrolyzed collagen, like the peptide, small peptide. When we conjugate with the polyphenols, so they have high activities. So I will be showing this slide example as example, so you can see that this is the wound healing activity. So when this is the control, this is like a cell, and these are the two cells, large number of cells. So we make a cut here. So here you can see that no cell is here. So when we added with the EGCG, a polyphenol, so you can see that this gap is filling. This because this cell will grow out to this side. You can see that. So this is how, like when we we got some kind of any injury. So once you start eating hydrolyzed collagen or plant polyphenols, you can see that they can improve your wound within at a higher rate. So they can fill your wound at higher rate. So I think you guys can understand this one, how it is happening. So once these cell will move, this is our injury. We made an injury here. So these cell will move toward this side when we add our hydrolyzed collagen or hydrolyzed collagen conjugated with the EGCG. So this is all about like our research that we have done here. And then next I will be about our center. So my student will be making a video of about for you, and you can see. So stop sharing. Hello, Mr. Ajay, can you hear me?
Yes, sir, I can hear you. Yeah.
Yeah. So can you please show the video?
Sir, can you make me a co-host? Because I am not host, I cannot hear.
Huh? Hello. Hello.
हेलो हाँ जी सची सर प्लीज मीट अजय
गेट खोल दो बस सारे। हाँ बस कुछ नहीं करना और लाइट जला दो। हेलो।
Yes.
नहीं भी वाला ऊपर जरा जाके क्या क्या हो गया आपका। हेलो।
Yes, I can hear you.
Sir, I am not. Can you make a co?
Sir, we have said that they are making you shout.
Ajay sir, you are now co-host.
I think if it is taking long,
Yeah. So this is our center, like International Center of Excellence in Seafood Science and Innovation. I think you can move to the instrumentation area. So these are the working spaces for the students. So we have almost 12 students in which around 4 are Indian as well as some Thai students and students from Indonesia. So these are the working space for the students. So currently we have received one funding that is called Reinventing University. So under which we are getting 12 students from all from and we so then that we you can. So we have so like we said that I said that we do the preservation of the seafood. So preservation, for that we need to use some technology, non-thermal technology that can enhance the shelf life of those foods for the longer term time. So these are the some of instruments. As you can see that this is the big one, the pulse electric field. So it can generate a pore inside the shrimp shell or other things, so that can allow their penetration into the cell, any compound, polyphenols or other compounds. And this is a kind of like cold plasma. So it inhibit the microbial growth. It is used for the like mostly packaged food. So once you pack the food, you can treat it using this technology. So they can almost we have stored the sample, fish sample, for almost 21 days using this technology in combination with the plant polyphenols. Yeah, this that's it. So and this is our like wet area where we usually use the cutting of the fish and spray. So we these are so this is all like for the pilot production. And you can see that this oxalate operator, so evaporator, rotary evaporator, that we use for the evaporation of the solvent from the plant extract. So we extract those polyphenols using ethanol. So you have to remove ethanol from those compound. So these are the instruments that are we can use to remove ethanol from the extract, and then you will get extract that have no ethanol it inside it, so that's easy to incorporate in any food. Yeah, so I think the time is over. Is it enough, ma'am? We have like one more lab about where we do cell culture lab that I show about wound healing. So the time is over, ma'am. Please, if it's
Sir, if there is something interesting to show,
sorry, ma'am.
if there is something interesting to share, then you can share, please.
Yeah. So this is a GC. I mean, so fish oil. So they have EPA, DHA. So this machine we use to determine that one. And next is for. Can you hello Ajay? I think his video is jam. Okay, so I will be I will be just showing the last slide. Just one. So that maybe is useful for the some students. So this is our activity recently. So we have receiving like 20 internship students from Gadwasu, Guru Angad Dev Veterinary University. So this is like we have funding that we can support some students. So 20 students from the bachelor, they will be coming here maybe in June for 2 week training. So just for the opportunity for any of your student who are willing to do like a master or maybe higher study for the PhD. So we have those funding and scholarships we can give. So like I already explained that we are receiving 12 students from various countries in Nigeria, Indonesia, and Bangladesh, Sri Lanka, and Ethiopia. So this is all we are doing here, and this is our like team of research student as well as our research lecturer. So yeah, this is all about for me, ma'am. So any question regarding the polyphenols? Please do not hesitate to ask me.
Girls, if you have any questions, please ask.
Good morning, sir.
Yeah, good morning.
Sir, this is Shruti Kalia from BSc Medical. Sir, in this lecture, we studied about the polyphenols and the phenomena, how we detect the presence in a particular compound. And we also saw the instruments. So, sir, I wanted to know that what is the basic standard time we need to actually extract the results, actually get the results from those instruments? Is it time consuming or is it instant result that we get from the instruments?
Uh, okay, I will. So for the analysis, it depends on the what kind of analysis do you want to do. So for example, this one. Sorry. So this is sorry. So this is all depend on the what kind of analysis you want to do. If you want to do antioxidant activity of this compound, for example, this is a dried bitter leaf powder. So if you want to determine its antioxidant activity, so there are 5 or 6 assays that we use to make sure that they have a proper antioxidant, because antioxidant is not related to just one free radicals. There are may be metagylating activity also. So it can take one day to determine the antioxidant activity for this one. For the microbial assay, and this one, okay, if you want to make a gel like this one, so it take almost 2 to 3 days, because you have to dry the sample. And for the instrumentation that we show to you, for the GC, for example, for one sample, it take around 2 hours. So it depends on how many sample you have. So sometime what we do, we run overnight. So we put like 6 or 7 sample, it will be run overnight.
Okay, sir. Thank you, sir.
Yeah, you're right.
Avtar sir, there is HPLC and FTIR also.
Yeah, it's okay, no problem. So there are some compound also. Okay, as I told you that these are this one is too messy. Okay, so what happened? These are you can see that those are amino group, NH group, or hydroxy group. We can determine using the FTIR, and for the HPLC we determine what kind of polyphenol is there using HPLC, like high. And for FTIR, you can know that what kind of protein is there, because you can determine this carboxyl group, carbonyl group, as well as the amide or amino group. So any more question?
Yes, sir.
Yes.
Sir, can you please tell us about the shelf life of these powders that we extracted from the polyphenols? How long we can use these powders?
Uh, we do not recommend like any compound to use like more than 3 months old, because if you extract this compound, they can easily in the presence of, if you don't keep them in the sunlight, if you in the dark, like these are the compound that should be present in the dark, because they have a hydroxyl group, they are easy to oxidize. So for us, we whenever we want to do one study, so we just use product that is prepared for lot like batch wise. So maybe I will not recommend maybe more than 1 month for this one, if you haven't kept in a -40 or under the dark conditions. But in this one encapsulation, okay, you can keep for the longer time, maybe 6 months, until 6 months you can keep.
And so, what about flavor change on these leaves?
Flavored for the leaves.
Yes, sir, flavor change on these leaves. Polyphenols. Using these polyphenols.
We do also sensory analysis. For example, if I show you this sushi or sashimi, so we do the sensory analysis, in which we take like around 30 to 40 students and ask them to taste, like look there by color. So they haven't found any change in the flavor for the first day, because these additives will not lead to any change in the color. That is our first priority, that any additive that you add onto the food, it doesn't change its appearance as well as the odor and flavor. So we always, but for polyphenols it is not the case. But when we use, like I talk about the hydrolyzed collagen, or sorry, this one hydrolyzed collagen. So sometime what happened, they have a fishy flavor, so they might affect the flavor or the taste of your food. But for the polyphenol, we haven't reported any change in the consumer acceptability. As you can see that this one, so you can see that at the day 0, the first 4, they always have almost similar appearance. So obviously there is no change when you add any extract.
Thank you, sir.
Yeah.
Excuse me, sir.
Yes.
Sir, right now you informed us that you give training to the bachelor students. So, sir, I wanted to know that what is the criteria you consider for selecting the students you want to train?
Criteria, actually this one is proposed by the university. So the students, those who want to, like, for example, you are a bachelor student, so you have 6-month training, 6-month research, maybe at the end of your semester. So if you want some of your study to done in our lab, like okay, you do some analysis in your, you do storage study, for example, you keep these polyphenol for 1 week or 18 days, and then you want to determine their the food, the fatty acid profile or FTR analysis you want to do in our lab, so you can come here and you can do. So there is no criteria, actually it should be related to our expertise, so you can come. There is no particular, like you should be high GPA or high percentage, but you should be hardworking. That's it.
Okay, sir. Thank you, sir.
Yeah.
Any other question? Any other query, girls? So I think there is no more queries. You explained each and everything very well, sir. Thank you so much, Dr. Avtar Singh, for joining us. And it's our privilege to have you as speaker, sir. The things are related to polyphenol and how they are helpful in conservation of food or increasing their shelf life. It was worth listening. And student must have understand the role of polyphenols and plants indeed, how they help in preservation of food material. I would like to express my deep gratitude to our worthy principal, ma'am, in absentia, for her unconditional support and motivation. I would also like to thank Professor Sadhna Tandon, Dean Life Sciences, for her guidance and moral support. I'm happy to express a vote of thanks to the whole faculty of Botany Department that has made this event a grand success through their motivation and dedication. I also want to thank Dr. Sandeep Singh and Dr. Sandeep Kaur for coordinating this event very well. So a special thank to Mr. Shashi from IT department for technical assistance, and to my students for being so interactive. Thank you so much for attending this
Thank,
webinar. Thank you, sir. Thanks
thank you
once
so
again.
much, ma'am, for inviting me to represent our work there. And I would like to thank to the principal, Professor Dr. Atima Sharma, and the HOD, Shikha Rashid. And also I would like to express my sincere thanks to Dr. Sandeep for considering me for this, to deliver a lecture to your institute. Thank you so much, everyone.
Thank you, sir. Thanks for joining us.